All right, good afternoon and hello. Welcome to class. We will begin today by talking about Watson-Crick hydrogen bonding. And this will be something I think you'll have seen before, but it will be important for you to recognize a correct Watson and Crick base pair versus something that looks like it but isn't exactly right. We talked about each of the individual bases last time. So let's just show you how they hydrogen bond with each other. So I'm going to copy the structure of the adenine base, and then we'll draw the thymine base or the uracil base that interacts with it. So we'll write down an A here. The exocyclic amino group of thymine interacts with a carbonyl oxygen from either the exocyclic base of adenine interacts with the carbonyl oxygen of thymine or uracil. Let's see if I can draw this angle. There we go. There's carbonyl oxygen. There we go. This is the attachment point to the sugar phosphate backbone. Then here is an NH bond. And if that's all we have, let's see, carbonyl oxygen there, then this lone pairs on this carbonyl oxygen hydrogen bond with the exocyclic amino group of adenine. And this NH bond, acting as a hydrogen bond donor, interacts with lone pairs on the nitrogen, the ring nitrogen from adenine. And those are the two hydrogen bonds that are present in a base pair between adenine and uracil. And this is, of course, a base pair that you would see in, say, RNA. If this were thymine, we would just copy everything we have here and then add a methyl group adjacent to the carbonyl right there, and then this becomes thymine. A couple of things to sort of keep yourself oriented. So this is an AT pair and an AU pair. The side of the base pair that has the squiggly lines indicating attachment to the sugar phosphate backbone, that's where the minor groove is. And so the other side, from here to here, whoops, I didn't mean to use that kind of arrow. From here to here is the major groove. And I'll show you a structure of double-stranded DNA so that you can get an idea. But if we were looking at, say, double-stranded base pairs in double-stranded oligonucleotides, that's where the major and the minor grooves would be. Some of you maybe are going to be uncomfortable with that because DNA is double-stranded, but RNA is not. So I'll put the major and minor groove stuff over by the DNA. Maybe I lost the E in groove. Okay. Man, struggling today. Okay. Questions about the AT base pair? One of the things you guys noticed last week was that adenine doesn't hydrogen bond with all of the possible groups on thymine or uracil. You've got this sort of unsatisfied carbonyl oxygen that's hanging out there, and that is true. The other thing to notice is that the major and the minor groove present different groups. For example, if you were looking at the major groove, the edge of this base pair, you would see a hydrogen bond acceptor, a hydrogen bond donor, and a hydrogen bond acceptor. And there's another lone pair, say, on that carbonyl oxygen that's available for interacting with other things. There's a hydrogen here, another lone pair there. So the major groove presents a constellation of hydrogen bond donors and acceptors, and transcription factors interact with those groups. So the minor groove also displays here, in this case, two hydrogen bond donors. Got it wrong. Electron donors, hydrogen bond acceptors. And there's a chemist at Caltech named Peter Dervin, who years ago invented a series of small molecules that would recognize specific base pair patterns in the minor groove, almost like synthetic transcription factors. Okay, so that's an AT base pair. We're going to repeat this for a GC base pair. So here we go. I'll just copy the G. And then we'll draw C over here on the right. Okay, so the carbonyl oxygen of the G base, hydrogen bonds with an exocyclic amino group on C. So we'll draw that here. All right, so here is the C base that we've just drawn. Here's a hydrogen bond donor group. Here's a hydrogen bond acceptor group, lone pairs on that ring nitrogen, and then here's another hydrogen bond acceptor group, lone pairs on that carbonyl oxygen. And you can see that a GC pair has 3 hydrogen bonds. The other thing you can notice if we compare, again, the difference between the major groove on this side, if this were in double-stranded DNA, and then the minor groove on this side, is that both major and minor grooves present distinct constellations of donor and acceptor types of atoms. So the pattern for a GC base pair in the major groove is hydrogen bond acceptor, acceptor, and then donor, whereas the pattern in the minor groove is acceptor, donor, acceptor. And again, transcription factors can interact with the edges of base pairs in this sort of specific way. We won't talk about that in any level of detail, but I want you to have the idea that even looking at the edge of a base pair, you can tell the difference between an AT pair versus a GC pair, and so on. Questions about that? Okay, so those are the bases. We need to talk about the rest of the structure of a nucleic acid. So there's a little bit of nomenclature to be aware of, and I do want you to know the difference between these two things. The term nucleoside refers to a base, which is sometimes called a nucleo or nucleobase, because they're found in the nucleus, I guess, plus ribose. So nucleoside refers to the N-glycoside of ribose with the nucleobase. So let's draw. Ribose is a sugar that you've no doubt heard of, but we haven't drawn in our chapter on carbohydrates. It's a 5-membered ring. I'm not going to attempt to draw it in a realistic conformation. We're just going to use a 5-membered ring. We'll use dashes and wedges. Correct stereochemistry at these 2 carbons is down or dash. Correct stereochemistry at carbon 5, where there's a CH2OH group, is up. The numbers of the ribose ring are numbered with primes to distinguish them from the numbering of the nucleobase, which nobody ever knows or pays attention to. But this is where we get the term 5' and 3' that you will have heard in your stuff that you've learned before about DNA. C1, carbon number 1', is the anomeric carbon. And that's where the nucleobase is going to be attached. And it's going to be attached via what we're going to call an N-glycoside. If this were an OR group, we would call that an acetal or a glycoside. If it's a nitrogen, we call it an N-glycoside. And let's just assume that this is the, say, adenine glycoside. So we'll draw the purine ring of adenine. There's a nitrogen here, a nitrogen there, double bonds, and then the exocyclic amino group, okay? So this is a nucleoside, and it's the nucleoside involving ribose and adenine. And so the naming of nucleosides is a little bit complicated. If the base is a purine, it's pretty easy. For A, we call this adenosine. So you have the aden from adenine. This is the adenine portion of the molecule. This is the ribose portion of the molecule. Maybe I'll highlight the adenine portion in green, and then maybe in orange, the ribose portion. So for the purine, we call the nucleoside just the sugar and the adenine, adenosine. So it's like adenine, but we put the os in the middle to indicate that it's the N-glycoside. If it's guanine, we do the same thing. We call the base guanosine. Now, I alluded to this last week, but for the other bases, it's a little bit trickier. If your base is a pyrimidine, the name can be challenging. So for you, if it's uracil, we just call this uridine instead of urosacil, because I guess that sounds stupid. If it's T, thymine, we call it thymidine. And if it's C, we call it cytidine. This is where you can get confused. If you're good at memorizing, and then this won't be a problem. If you're only familiar with memorizing, you're going to get confused because the name of C is cytosine. So if you memorize, if it sounds like osine, it's got ribose in it, you're going to get it wrong. I didn't make up the rules. They just chose them this way, unfortunately. Okay, so the base for C is cytosine. The nucleoside is cytidine. You may need to work on that and memorize it. I don't know, some mnemonic device. Good luck. Okay, questions about that? Now, those are ribonucleosides. And that is a structure that is found minus the phosphate part, but that's something that's on its way to be, sorry, I'm gonna sneeze, that's obnoxious. That's something that's on its way to be found in RNA. Deoxyribonucleotides are going to be exactly the same, but they're going to lack one of the oxygens of the ribose ring. So deoxyribonucleosides lack the 2' hydroxyl. And nature actually has to have a way to do that, starting from ribose, getting rid of that OH group. And we're not going to talk about that. It's a reduction reaction. But a deoxyribonucleoside is 2' deoxy, let's see, what's the right way to say this? 2' deoxy, meaning the oxygen is no longer there. 2' deoxyribose plus the base. And again, it's an N-glycoside because you have the base attached at the anomeric carbon via a nitrogen. You're going to name all of these deoxynucleosides exactly the same as you would name the corresponding nucleoside. When you put an abbreviation, you'll use DA to indicate it's 2' deoxy. And then in the name, you're just going to add deoxy in front of adenosine. So DA would be deoxyadenosine, DG would be deoxyguanosine, and so on. There's no DU because uracil is not used in DNA. So all you have to worry about here are DT and DC. Deoxythymidine, and then deoxycytidine. Now that 2' OH group plays an interesting and important role. If you've ever been in a molecular biology lab, you'll know that DNA is relatively stable, but RNA you have to be careful with, in part because there's ribonucleases or enzymes produced on your hands in your body that digest RNA pretty quickly. But also RNA has some intrinsic instability because of this 2' OH group that maybe we'll talk about in a minute. But yeah, go ahead.
So if you can have DT but not DU, shouldn't there not be a T in the ribonucleoside?
Should there or should there not be a T? I don't know. I think that's how you would name it if it were. If you want, though, we can erase it from the list. You've reached the limits of my biology knowledge, so good luck. Okay, what else? All right, so that's the sugar and base. We need to add a sugar phosphate. We need to add the phosphate portion. All right, so this is kind of like stalactites and stalagmites. You're going to need a mnemonic device to figure it out. Once you attach the phosphate portion, they are called nucleotides, as in ribonucleotides and deoxyribonucleotides. So ribonucleotides include the nucleoside plus a phosphate group. If you look at the ribose ring, there are many places where you might consider attaching a phosphate group. And a lot of the different varieties are known. I'm only going to show you the most important ones where we attach the phosphate to the 5' hydroxyl group via a phosphodiester. So we'll erase the hydrogen on that 5' OH group, and we'll attach the phosphate. So again, this is our ribose ring, and then this is the phosphate group in pink. And so the name of what we've just drawn here, we're going to keep the same name, only we're going to add a little bit to this. Adenosine, which takes care of the adenine and the ribose. Then we need to add which OH group the phosphate's on, because we have some choices. And then we have to indicate that there is just one phosphate on it, because you're going to see that we can often have more than one phosphate attached over here. So what we've just drawn for you is the structure of adenosine 5' monophosphate, or AMP, and this is an example of a nucleotide. And then if we wanted, we could add an additional phosphate, and you've seen the structure of ATP before, right? If we attach another phosphate here, that's ADP, right? And if we attach another phosphate here, that's ATP. And we talked before about these individual phosphates. We talked about phosphoanhydride bonds before. We've talked about phosphate groups as leaving groups. So hopefully you can remember what we talked about before in that area, and how these phosphate oxygen bonds are relatively weak and subject to nucleophilic attack. We saw before that sometimes a nucleophile would attack at this terminal phosphate, whereas other times a nucleophile would attack at this internal phosphate, and that regioselectivity would really depend on the enzyme that's catalyzing the reaction. Okay, questions about that? Okay, some of this will be new. Go ahead.
Do you ever see like the acyclic version of the ribose in nature?
Do you see the acyclic ring-opened version of the ribose in nature? Not with DNA or RNA.
Also, how good of a leaving group is the nitrogenous base?
How good of a leaving group is the base? There are enzymes that can, proofreading enzymes that will go in and determine a base that shouldn't be there, clip it out, and put another one in. So it's decent enough that enzymes can make it happen. Presumably that would involve probably some kind of proton transfer at the same time as electrons on the oxygen kick down and kick off the leaving group. Okay, good. Others? All right. Well, let's do just briefly a mechanism that's relevant for oligonucleotide synthesis in your body. So this is how you take one nucleotide and attach another to start making a strand of DNA or RNA. We'll do this for now with DNA. So let's just suppose that we have deoxyadenosine monophosphate. So what's that going to look like? Actually, let's make it G, so we have to practice drawing something other than adenine. Let's see how you are at drawing this. So here's the phosphate. There should be an OH group on the 3' carbon. There should not be an OH group on the 2' carbon. And actually, let's suppose that this is deoxyguanosine 5' monophosphate. I guess that was implied in how I wrote it out. There's a phosphate here. Okay, and then we're going to draw the structure of G. We could just abbreviate it with the G because it's not going to participate in the reaction. It won't be relevant for the mechanism, but it's decent practice for you to work on drawing the structure of the guanine base versus the adenine base versus the other bases that we'll learn about. So there's the G base. And let's suppose we want to make a dinucleotide. Sorry, we need to add page. And the way nature will do this is it will use a nucleotide triphosphate as the electrophile. And it will use the 3' OH group of another nucleotide as the nucleophile. Okay, so lone pairs on that oxygen are going to be important. Let's draw ATP really quickly. Again, the ribose ring. You're going to get sick of doing this, but you should practice it because in the fog of war on an exam, sometimes these start to all look pretty familiar. So it's good to have a sufficient amount of practice. Okay, that's the adenine base. This is 2' deoxyadenosine 5' triphosphate. So here's the 5' hydroxyl. There's the 3' hydroxyl. On carbon 2', there is no OH group. Then we'll draw the 3 phosphates of ATP, remembering all the while that these are phosphoanhydride bonds that are about as reactive as a carbon-based anhydride and subject to sort of the same nucleophilic addition followed by elimination types of reactions that carbonyls are. All right. That's ATP. Everything look okay there? Aside from the penmanship? All right. So, an enzyme is going to first deprotonate the 3' hydroxyl group on deoxyguanosine 5 monophosphate, or DGMP. Now, I don't know about you, but I am not excited about drawing that proton transfer step and redrawing everything. So, if it's okay, we're going to imagine what that would look like. it would look like some basic group from the enzyme removing the proton to give you a negatively charged oxygen. We've seen examples before of how bases in enzyme active sites can remove protons, so that shouldn't be that surprising. Then the 3' hydroxy group becomes a 3' alkoxide, which is now a good enough nucleophile to attack the phosphate, and we're going to attack the internal one. Now, off the top of my head, I forget. I remember we called them alpha, beta, and gamma, but I can't remember whether we started alpha at the 5' hydroxy or we started at the end. It doesn't really matter. We'll call it the inside one, okay? You'll learn in biochemistry which one's alpha and which one's gamma, but for now, since I forgot, let's have the oxygen attack that internal phosphorus. And just like in carbonyl chemistry, electrons from that carbon, phosphorus, oxygen pi bond will kick up onto the oxygen to give us an intermediate. Now I know you're excited about drawing that. So let me show you where you can abbreviate. Let's draw that intermediate. The phosphorus should be bonded to 5 things now. One is the group, the oxygen that used to be doubly bonded to the phosphorus. That's sort of like your carbonyl oxygen. One is the 5' hydroxyl of the ribose ring, or the deoxyribose ring. At this point, we're just going to draw A. Can you live with that?
Sure.
Probably, yeah. Okay. All right, and then the next thing is this negatively charged oxygen here. The next thing after that is these 2 phosphorus groups that are linked to each other. So we'll just draw that here. This is sort of 2 phosphate groups linked to each other through a phosphoanhydride bond, are basically considered a, you could call this pyrophosphate. And this is ultimately going to be a, except you can't spell when you're pyrophosphate. It's basically going to be a leaving group in what happens next. Biochemical texts will abbreviate that as 2 P's with a subscript I, meaning pyrophosphate. And the I actually stands for inorganic. And why they call it that is because there's no carbons in the molecule. Lastly, we have the new bond that we just made, which I'll highlight in red. And that's the bond to the 3' hydroxy group of the DGMP base that we used as our nucleophile. So we'll draw the rest of that here. We'll put the G here. There's no oxygen on the 2' hydroxyl. And then on the 5' hydroxyl, we have a single phosphate group. Okay. So take a look at that real briefly and make sure I didn't make any mistakes. See anything? Yeah.
You just spelled pyrophosphate. You forgot the S.
I spelled pyrophosphate wrong, and you thought I would want to know about that. I did ask, didn't I? Okay. As my father used to say, you have obviously mistaken me for someone who cares. No, I do care about that. Thank you. What else? Okay, so what we're going to do now is basically, and you should try to see the connection between this and all the carbonyl chemistry we've done previously. Electrons kick down and leaving group leaves. All right? And then we're going to draw what we just, we're going to draw the product. And I'm not going to draw the individual bases anymore, but I do want to draw everything else so you can see pretty clearly what we've made. And this will hopefully help you understand some of the language you've heard in the past around oligonucleotides. So here's the 3' hydroxyl group. It's got a bond to a phosphorus. And then that phosphorus itself is bonded to the 5' hydroxyl group of another unit. And that's A. Okay. Also, a product is inorganic pyrophosphate, and that itself, if you add water, can be hydrolyzed into 2 molecules of inorganic phosphate, where that's just one of the conjugate bases of phosphoric acid, either the fully deprotonated one or probably the mono phosphate version. Okay, and anytime pyrophosphate is a leaving group in a reaction, probably this is also happening downstream, and that actually adds some thermodynamic driving force for these reactions to happen. Now the 2 sugars are linked together, and this type of linkage, if we have just one phosphorus group on an oxygen, that is called a phosphodiester. That's this bond right here. If we have 2 OR groups linked to a phosphate, we'll call that functional group a phosphodiester. So I know you've heard that term before. Somebody's referred to the phosphodiester linkage between nucleotide units in a oligonucleotide. And so DNA and RNA consists of phosphodiester sugar, and then you could see how we could elongate this and put another nucleotide on there, right? Using this 3' hydroxyl as a nucleophile, we could extend and extend and extend. Okay? So this is how DNA and RNA replication works. Always the 3' hydroxyl is the nucleophile, and always a nucleotide triphosphate is the electrophile. And always you're making phosphodiesters. See your hand? Yeah, go ahead.
I was just going to ask, so it's always a triphosphate, but you can't do like a diphosphate?
Right. Does nature always use triphosphates? As far as I can tell, yes. Presumably it could be done with ADP, but possibly the reason is you need this extra driving force. Okay. We haven't talked about how nature knows how to put an A here bonded to the G instead of something else. That has to do with the molecular recognition, hydrogen bonding associated with the different base pairs. But there's more to that story, and unfortunately we don't have time to talk about it. Okay, other questions about nucleotide structure? Yeah?
So is that what are you talking, like hydrolysis?
Yeah, that's hydrolysis of pyrophosphate, yep. And you could draw that mechanism very similar to how we drew this mechanism. The oxygen of water would attack one of the phosphoruses of the pyrophosphate. You get a pentavalent intermediate, electrons would kick down, and the leaving group would leave. Okay. One thing that's maybe worth noting is why RNA is somewhat less stable than DNA, and it's not that hard to understand. Let's just pretend for a moment we'll draw on this molecule. This is a dinucleotide. We'll draw in temporarily a 2' hydroxyl group. Now, under basic conditions, you could imagine this 2' hydroxyl group getting deprotonated. And then you have a phosphorus atom that's electron poor because it's bonded to 4 oxygens, and you've got a nucleophile really close to it. That oxygen can attack the phosphorus, and you can actually break one of these diester linkages. And so RNA sometimes is, you have to be careful with basic conditions around RNA. The presence of that 2' hydroxy group is basically introduces an internal nucleophile that can break apart the phosphodiester linkage. There's more to say about that, but I'll perhaps leave that for your biochemistry class. Lastly, I think I just want to show you real briefly some things about overall DNA structure. I think we've already done everything that would need to be done in this particular diagram that's straight from your text. Note that the 5' end of DNA always has a phosphate on it. Note that the 3' end of DNA is always the OH group. That's where you can attach additional nucleotides. Notice the sugar phosphate backbone always 3' to 5'. DNA is synthesized 5' to 3' for the reasons that we've just talked about. Then individual strands of DNA can associate via hydrogen bonding between the bases to give this double-stranded form of DNA. And here is a space-filling model of that. And then here is a cartoon of that. Interestingly, in DNA, the two strands assemble anti-parallel to each other. So you have one strand, here's the 5' end that would have the phosphate group, and following it down, here's the 3' end that would have the OH group. And then the other strand assembles anti-parallel to it with 5' phosphate here, cycling upward to a 3' OH group. And at each step, the bases are complementary to each other. So you can design an oligonucleotide strand that will bind a specific other oligonucleotide strand just based on sequence alone. There are, as you can see from double-stranded DNA, there's places where the sugar phosphate backbone, you can tell that here because you've got alternating phosphate atoms that are yellow, oxygens that are red, and then sugar ribose that's carbon is gray and hydrogen is white. So the sugar phosphate backbone of one strand is here. And you can tell that it's pretty close to the sugar phosphate backbone of the other strand. That close spacing is called the minor groove. It's a groove because it indents inward, minor because it's smaller. This larger distance is called the major groove, and we've referred to where those are in the individual bases. As you look at the edge of the individual bases, you can see some of those donor-acceptor patterns like we saw earlier. Notice hydrogen bond acceptor, hydrogen bond donor, hydrogen bond acceptor. So I want you to go back in your notes, look at the major groove edge of a Watson-Crick base pair, and then I want you to tell me, based on that, what type of base pair is that? You have to think about it. Transcription factors don't. They can tell. Go back. Look at the two different base pairs of DNA, an AT base pair versus a GC base pair. Think about where the major groove is and see if you can tell me for this one here, I'll circle it, I'll point an arrow to it, what kind of base pair is that? Oh, dang, I just invented an excellent exam question, is what I just did. Just so you know, by the way, in this space-filling model, nitrogens are blue, oxygens are red. That's going to be backwards from the color scheme I chose in our drawing, so good luck with that. Sorry. It might help. This oxygen here is a hydrogen bond acceptor. This NH is a hydrogen bond donor. This nitrogen is a hydrogen bond acceptor. ADA is the pattern. What base pair is it? Some of you are saying AT, and that's right. Let's copy. We'll see if we can get it by. I'm going to go back to the notes. Really, the choice of blue and red for hydrogen bond donors and acceptors may have been unfortunate because whatever. Paste it here. Oops, no. What happened? Dang it. Nope. Paste, shrink, drag. Better. Okay, we're looking in the major grooves. So we would be looking here. You have a pattern oxygen acceptor, NH donor, nitrogen acceptor. There's your oxygen over here, acceptor, nitrogen donor, nitrogen acceptor. See it? Sort of. No. Okay, go ahead.
How can you tell it was an acceptor, a donor, and then an acceptor?
From which drawing? Are you okay with that here but not here?
Correct. Yeah.
Okay. Okay, good. So I looked at this red atom. It's an oxygen and there's no hydrogen attached to it. So therefore that has to be a carbonyl oxygen, right? The next thing I see is this blue nitrogen, oops, and it's bonded to a hydrogen. Okay, the white is hydrogen, blues are nitrogen, red is oxygen. And then I see here a nitrogen and there's no hydrogen on it. So I infer that this carbon, that oxygen is a carbonyl oxygen with a lone pair. I infer that that's a ring nitrogen with a lone pair. I infer that this is an exocyclic nitrogen with an NH group on it. Okay? So you see the pattern there of acceptor, donor, acceptor, and that matches an AT pair. Okay? So good. Should we do another one? Since I just closed, now that we have a fun exam question? I'm laughing, but you're not. Okay. Let's look at the next one up. So now we're going to be looking at this one. Let's walk through it together. This is an NH group. This is an oxygen, and this is a nitrogen. So the pattern there is donor, acceptor, acceptor. So now let's go back and scroll up and look at the Watson-Crick base pair for GC and see whether that matches what we expect. We already know what to expect for the major groove of AT. So let's see what we can expect here. For the major groove of GC, we expect to see a donor, or sorry, got it wrong, a hydrogen bond acceptor, a hydrogen bond acceptor, and then a hydrogen bond donor. And that is indeed what we see, donor, acceptor, acceptor. That tells you, by the way, if this is a GC base pair, which side is G and which side is C? Left is C or G? Which one has the exocyclic amino group? That's C. And then which side has the oxygen and the nitrogen? That's G. Okay, good. Let's do the same for the AT base pair. Which side has the exocyclic NH for an AT base pair? That's A. And then, so that must mean this is T. And actually, there's your methyl group for T. The methyl group for T sticks out into the major groove. Okay? Yeah?
Did you tell us that was major groove, or should we be able to infer that?
I told you it was the major groove, but it's easy. You just look at distances. This is a small distance. That's a larger distance. The bigger distance between sugar phosphate backbones is the major groove. Now, I probably am not going to ask this question about the minor groove because it's harder to see into the minor groove. But I suppose if I gave you a double-stranded DNA structure and I illustrated it like this, I could ask you which of these levels is an AT base pair versus a GC base pair. Maybe that's not a good question, though, because it's 50-50, isn't it? Oh, no, you got 4 combinations, right? It could be AT, TA, GC, or CG. That's a decent question. Do it. Hey, I may be evil, like Dr. Doofenshmirtz, but at least I disclose my evil ahead of time. Okay, questions about that? That was fun. All right. Oh man, it was so fun. It's now 7:45 in Sydney, and I didn't even notice. Okay, so we're now going to move on to our chapter about metabolism. And we're going to be describing, basically, I think probably the correct term is catabolism. This is converting biomolecules into smaller bits and extracting energy along the way. We will focus much of our time on carbohydrates. We're actually going to start a bit with lipids. We'll do carbohydrates in the most detail, and proteins we will basically leave for biochemistry. So what you need to know about carbohydrates is the process of going from glucose through something that's called glycolysis, or sugar lysis, or breaking of sugars, to a molecule that's a 3-carbon chain called pyruvate. Okay, that's an important intermediate. By the way, glucose has 6 carbons, so the fact that we're breaking glucose up into pyruvate tells you must be breaking it into 2 pyruvates. And that's glycolysis. That process happens in the cytoplasm. Then nature isn't done there. There's a process called oxidative decarboxylation. Some of you wanted to say phosphorylation. We're not actually going to do oxidative phosphorylation. That's something for your biochemistry texts. There we lose CO2, and we convert pyruvate from pyruvate into acetyl-CoA, which you've seen before when we learned about thioesters. Acetyl-CoA ends up getting put into something called the citric acid cycle, sometimes also called the tricarboxylic acid cycle, sometimes also called the Krebs cycle. We'll learn about that in a lot of detail. CO2 is spit out of the Krebs cycle, but you also get GTP, and you also get some energy-rich molecules like NADH and FADH2, and then those go to the electron transport chain in mitochondria, where they participate in oxidative phosphorylation to reduce oxygen to water and to generate ATP. Okay, so that's the downstream flow of events for carbohydrates. Lipids, will start with triglycerides, and we'll talk about how lipids hydrolyze the triglyceride into fatty acids and glycerol, but we'll see that lipids intersect both with glycolysis, because the glycerol portion of the lipid can intersect with glycolysis, and then the fatty acid portion is going to get converted into acetyl-CoA. I'll fill in a little bit of detail next time with proteins, but for the most part, we will ignore amino acid metabolism. I guess that's enough for today. I'll see you next time.